ಗುಡ್ ಆಫ್ಟರ್ನೂನ್. ಬಿ ಯು ಸಿ ಸೆಕೆಂಡ್ ಇಯರ್ ಬಯಾಲಜಿ ಪಾಸಿಂಗ್ ಪ್ಯಾಕೇಜನ್ನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಹಾಗಾದರೆ 12th ಚಾಪ್ಟರ್ ಏನಪ್ಪ ಅಂದರೆ ಅಪ್ಲಿಕೇಶನ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ, ಹೌದಾ? ಜೈವಿಕ ತಂತ್ರಜ್ಞಾನದ ಅನ್ವಯಗಳು. ಹಾಗಾದರೆ ಮೆನ್ಷನ್ ಅ ಜೀನ್ ದ್ಯಾಟ್ ಕೋಡ್ಸ್ ಫಾರ್ ವಟ್ ಇನ್ಸೆಕ್ಟಿಸೈಡಲ್ ಪ್ರೋಟೀನ್ ಇನ್ ಬಿ ಟಿ ಕಾಟನ್ ಬಯೋಟೆಕ್ನಾಲಜಿಕಲ್ ಕಾಟನ್. ಓಕೆ, ಅಲ್ಲೇನಪ್ಪ ಅಂದರೆ ನಾವು 3 ಜೀನ್ಸನ್ನು ಕಾಣ್ತೀವಿ, ಹೌದಾ? ಎಷ್ಟು 3 ಜೀನ್ಸನ್ನು ಕಾಣ್ತೀವಿ. ಒಂದು ಕ್ರೈ 1AC, ಕ್ರೈ 2AB, ಆಮೇಲೆ ಕ್ರೈ ಜೀನ್ ಅಂತ ಬರ್ತದೆ, ಹೌದಾ? 3 ಸೇಮ್ ಆಲ್ಟರ್ನೇಟಿವ್ಲಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಆಮೇಲೆ ವಾಟ್ ಈಸ್ ಅ ಬಯೋಪೈರಸಿ? ಬಯೋಪೈರಸಿ ಇಸ್ ದ ಟರ್ಮ್ ಯೂಸ್ ಟು ರೆಫರ್ use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. ಹೌದಾ? ಇದನ್ನು ಬಯೋಪೈರಸಿ ಅಂತ ಕರಿತೀವಿ. ವಾಟ್ ಈಸ್ IRNA? ಹೌದಾ? IRNA RNA ಇಂಟರ್ಫೆರೆನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. IRNA ಕರಿತೀವಿ, RNA ಇಂಟರ್ಫೆರೆನ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಇನ್ ಆಲ್ ಯೂಕ್ಯಾರಿಯೋಟ್ ಇಟ್ ಈಸ್ ಅ ಮೆಥಡ್ ಆಫ್ ಸೆಲ್ಯುಲಾರ್ ಡಿಫೆನ್ಸ್. This method involves silencing of a specific mRNA due to complementary double-strand RNA molecule that binds and prevent translation of the mRNA. Okay, mRNA, modified RNA. What are genetically modified organisms? GMO ಅಂತ ಕರಿತೀವಿ ಅದನ್ನು. Genetically modified organisms, GMO. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms. Next, 2 more questions ನೋಡೋಣ. Mention any 2 critical research areas of biotechnology. Providing the best catalyst in the form of improved organism using a microbe or pure enzyme. Second one, creating optimal conditions through engineering for the catalyst to act. Third one, downstream processing technology to purify the protein organic compound. Okay, next question, what is gene therapy? Name the disorder to which what clinical gene therapy was given first. ಹೌದಾ? ಜೀನ್ ಥೆರಪಿ ಏನಪ್ಪ ಅಂದರೆ ಅನುವಂಶೀಯ ಚಿಕಿತ್ಸೆ ಅಂತ ಕರೀಬೋದು, ವಂಶವಾಹಿ ಚಿಕಿತ್ಸೆ, ಹೌದಾ? ಆಮೇಲೆ ನೇಮ್ ದ ಡಿಸಾರ್ಡರ್ ವಿಚ್ ಕ್ಲಿನಿಕಲಿ ಜೀನ್ ಥೆರಪಿ ವಾಸ್ ಗಿವನ್ ಫಸ್ಟ್. ಹೌದಾ? ಈ ವಂಶವಾಹಿ ಚಿಕಿತ್ಸೆಯನ್ನು ಕೊಡೋದ್ರಿಂದ ಉಂಟಾದಂಥ ಸಮಸ್ಯೆ ಕೇಳಿದೆ. ಜೀನ್ ಥೆರಪಿ ಈಸ್ ದ ರಿಪ್ಲೇಸ್ಮೆಂಟ್ ಆಫ್ ಅ ಫಾಲ್ಟಿ ಜೀನ್ ಬೈ ಎ ಹೆಲ್ದಿ ಆ್ಯಂಡ್ ಫಂಕ್ಷನಲ್ ಜೀನ್. ಓಕೆ, ADA, adenosine deaminase deficiency. ADA, it's a disorder. 3 more questions. How is ADA deficiency cured by gene therapy? A lymphocyte from blood of the patient are grown in a culture ಔಟ್ಸೈಡ್ ದ ಬಾಡಿ. ಏನು ಮಾಡ್ತಾರೆ ಅಂದರೆ ನಮ್ಮ ಬಾಡಿ ಲಿಂಫೋಸೈಟ್ಸನ್ನು ಔಟ್ಸೈಡ್ ದ ಬಾಡಿ ತೆಗೆದುಕೊಂಡು ಕಲ್ಚರ್ ಮೀಡಿಯಮಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡ್ತಾರೆ. ಆ ಫಂಕ್ಷನಲ್ ADA, ಓಕೆ, ಆ ಫಂಕ್ಷನಲ್ ADA ಬಟ್ ಅಡೆನೋಸಿನ್ ಡಿಅಮೈನೇಸ್ ಡೆಫಿಷಿಯನ್ಸಿ, a functional ADA and cDNA is introduced into these lymphocytes. ಇವುಗಳಿಗೆ ಏನು ಮಾಡ್ತಾರೆ, ಎರಡನ್ನು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಂಥ ಕೆಲಸ ಮಾಡ್ತಾರೆ ಯೂಸಿಂಗ್ ಅ ರೆಟ್ರೋವೈರಲ್ ವ್ಯಾಕ್ಟರ್. ಆ್ಯಂಡ್ ದ ಲಿಂಫೋಸೈಟ್ಸ್ ಆರ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ಡ್ ಬ್ಯಾಕ್ ಇನ್ ಟು ದ ಪೇಷಂಟ್. ಮತ್ತು ಅವುಗಳನ್ನು ಏನು ಕಲ್ಚರ್ ಮೀಡಿಯಮಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡಿರ್ತಾರಲ್ಲ ಅದನ್ನು ನಮ್ಮ ದೇಹದ ಲಿಂಫೋಸೈಟ್ಗಳಿಗೆ ಹಾಕುವಂಥ ಕೆಲಸ ಮಾಡ್ತಾರೆ. ಪೀರಿಯಾಡಿಕ್ ಇನ್ಫ್ಯೂಷನ್ ಆಫ್ ಸಚ್ ಅ ಜೆನೆಟಿಕಲಿ ಇಂಜಿನಿಯರ್ಡ್ ಲಿಂಫೋಸೈಟ್ಸ್. ಹೌದಾ? ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಒಂದಾದ ನಂತರ ಒಂದು ಹಲವಾರು ಸಲ ಪೀರಿಯಾಡಿಕ್ ಇನ್ಫ್ಯೂಷನ್ ಆಫ್ ಸಚ್ ಜೆನೆಟಿಕಲಿ ಇಂಜಿನಿಯರ್ಡ್ ಲಿಂಫೋಸೈಟ್ ಈಸ್ ಗಿವನ್ ಆ್ಯಸ್ ದೀಸ್ ಸೆಲ್ಸ್ ಆರ್ ನಾಟ್ ಮಾರ್ಟಲ್. ಅವು ನಮ್ಮ ದೇಹದಲ್ಲಿ ಸಾಯದೇ ಉಳಿದ್ರೆ, ಇಫ್ ದ ಜೀನ್ ಐಸೋಲೇಟ್ ಫ್ರಮ್ ದ ಮ್ಯಾರೋ ಸೆಲ್ಸ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್, ನಾವೇನು ಜೀನ್ ಐಸೋಲೇಟ್ ಮಾಡಿರ್ತೀವಲ್ಲ ಅದು ಮ್ಯಾರೋ ಸೆಲ್ಸಿಂದ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ಲಿಕ್ಕೆ ಪ್ರಾರಂಭಿಸಿದ್ರೆ ADA ಈಸ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ಡ್ ಇನ್ ಟು ಸೆಲ್ಸ್ ಎಟ್ ಅರ್ಲಿ ಎಂಬ್ರಿಯಾನಿಕ್ ಸ್ಟೇಜ್. ಇಟ್ ಕುಡ್ ಬಿ ಪರ್ಮನೆಂಟ್ ಕ್ಯೂರ್. ನಾವು ಅದನ್ನು ಮಾಡ್ಬೋದು, ADAನ ಕ್ಯೂರ್ ಮಾಡ್ಬೋದು. ಇನ್ ವಿಚ್ ವೇಸ್ genetically modified plants are useful? Genetically modified crops more tolerant to abiotic stresses. Abiotic stresses are called what cold, drought, salt, heat. Genetically modified crops what reduced what reliance on the chemical pesticides, pest resistant crops. Genetically modified crops help to reduce post harvest losses. ಹೌದಾ ಆಮೇಲೆ ಜೆನೆಟಿಕಲ್ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಇನ್ಕ್ರೀಸ್ ದ ಎಫಿಷಿಯನ್ಸಿ ಆಫ್ ಮಿನರಲ್ ಯೂಸೇಜ್ ಬೈ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, ದಿಸ್ ಪ್ರಿವೆಂಟ್ಸ್ early what exhaustion of fertility of the soil. Genetically modified crops enhance the nutritional value of food, example vitamin A enriched rice. Okay, what is a biopiracy? Explain if with reference to basmati rice. B question, what are genetically modified organisms? Name two biotechnological toxin producing plant ಅಂತ 5 marks, 3 marks ಮತ್ತು 2 marks, total 5 marks ಒಂದು ಕ್ವಶನ್ ಇದೆ. Then biopiracy is a term used to refer bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Basmati rice is the distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. In 1997, an American company got a patent rights on basmati rice through the US Patent and Trademark Office. This new variety of a basmati had actually been derived from Indian farmers' varieties. In what Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or what a novelty. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms (GMO). Bt toxin producing plants are Bt cotton, Bt corn, rice, tomato, potato, and soybean. These are what biotechnological toxin producing plants: cotton, corn, rice, tomato, potato, and soybean, etc. One of the application of biotechnology One of the applications of biotechnology is the production of insect resistant crop plants. Justify the statement with reference to Bt cotton. The cry gene, cry1AC, cry2AB from Bacillus thuringiensis coding for the toxins are transferred to cotton plants. The protein encoded by these genes control the bollworms because the toxin itself what produced in a plant body. This Bt cotton plant expresses the Bt toxin gene by producing the toxin that provides insect resistance. Bt toxin is a toxic insecticidal protein formed in the form of protein crystals that kills certain insects. It exists as a inactive toxin, and once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activated toxin binds to the surface of epithelial cells of the midgut and creates pores, which cause cell swelling and lysis, and eventually the death of insect occurs. What is a gene therapy? Give an example. Write a note on biopiracy with reference to basmati rice. A replacement of a faulty gene by a healthy and functional gene is called as a gene therapy. Example, the first clinical trial on gene therapy was attempted on 14th August 1990 on a young girl suffering from genetic disease called ADA, adenosine deaminase deficiency. Then what they ask, write a note on biopiracy. Biopiracy is the term used to refer, use or exploit of a patent of a biological resource by multinational companies and other organizations without proper authorization from the uh, countries and the people concerned without compensatory payment. Basmati rice. In 1997, an American company got the patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell new variety of basmati in US and abroad. This new variety of basmati had actually been derived from Indian farmer varieties Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Then, development of a BT cotton has reduced the use of insecticides. Justify. Write a note on gene therapy. Same repeated question. Some strains of a Bacillus thuringiensis produce a protein that kills certain insects. They are called cry proteins. The genes which produce them are called cry genes. They are incorporated in the cotton plant by rDNA technology. When insect and pests like bollworms feed a cotton plant, they are killed. Thus, the development of BT cotton has reduced insecticides. Gene therapy, the technique of replacing a defective gene by a normal gene to correct a genetic disorder. Adenosine deaminase deficiency is due to deletion of the gene from adenosine deaminase. This can be cured by gene therapy, hence immunity is restored. What is RNA interference? How does RNA interference helps to develop resistance in tobacco plant against a nematode infection? The silencing of specific mRNA due to complementary double-strand RNA molecule that binds and prevent translation of mRNA is called RNA interference. A nematode meloidzine incognitia infects the roots of the tobacco. Meloidzine incognitia infects the roots of a tobacco plant and reduces their yield. Nematode specific gene were introduced into the host plant using agrobacterium vectors. The introduced DNA produce both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded RNA dsRNA that initiates RNA I and silenced the specific mRNA of the nematode. The result was that the parasite could not survive in a transgenic host and the transgenic plant got itself protected from the parasite. What is a biopiracy? How ADA deficiency is cured by gene therapy? Repeated question again. Biopiracy is the term used to refer to use or exploit of a patent of a biological resource by multinational companies and other organizations without proper authorization from the countries and the people concerned without compensatory payment. Then lymphocytes. Lymphocytes from a blood, ADA, the asked deficiency. Lymphocytes from the blood of the patient are grown in a culture outside the body. Then functional ADA and cDNA is introduced into these lymphocytes using a retroviral vector, and the lymphocytes are introduced back into patient. Periodic infusion of such a genetically engineered lymphocyte is given as these cells are not immortal. If the gene isolate from the bone marrow cells producing ADA is introduced into cell at early embryonic stages, it could be a permanent cure. List the benefits of a transgenic animals. Study of a normal physiology and development. Specifically designed transgenic animals are used to study gene regulation and its role in the normal functions of the body and its development. Study of diseases. Specifically designed transgenic animals are used as a models to understand the role of genes in development of diseases like cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Production of biological products. Transgenic animals are designed to produce useful biological products alpha 1 antitrypsin and milk enriched with alpha uh, lactal lactalbumin that is used in medicines required for the treatment of disease. Okay. Next, testing of a vaccine safety. Transgenic mice which can replace monkeys are being developed for use in testing the safety of vaccines like a polio vaccine before they are used on humans. Testing of a chemical safety or toxicity safety testing. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. they are then exposed to the toxic substances, and the effects are studied. Okay, up to now we discussed what a chapter. What is a chapter? Applications of biotechnology. Applications of biotechnology. Important terms we discussed. Whatever terms we discussed here, 1 mark question, 2 mark questions, 3 mark questions, maximum. Whatever the questions came repeated, like a biopiracy, then GMO, or whatever the other questions are there. Okay, repeated questions are there, they maximum come in our ಎಕ್ಸಾಮ್ಸ್. ಅದಕ್ಕೆ ಏನು ಮಾಡಿರಿ, ಯಾವ ರಿಪೀಟ್ ಕ್ವಶನ್ ಹೆಚ್ಚಾಗಿದ್ದಾವೆ ಅವುಗಳನ್ನ ಹೆಚ್ಚು ಸ್ಟ್ರೆಸ್ ಮಾಡಿ ಓದುವಂಥ ಕೆಲಸ ಮಾಡಿರಿ. ಆಮೇಲೆ ರಿಪೀಟ್ ಯಾಕೆ ಮಾಡಿದಾರಪ್ಪ ಅಂದರೆ ಅಲ್ಲಿ ಒಂದು ಸೈಕಾಲಜಿಕಲ್ ಟರ್ಮ್ ಇದೆ, ರಿಪೀಟೆಡಾಗಿ ನೀವು ಕೇಳೋದ್ರಿಂದ ಅಥವಾ ರಿಪೀಟೆಡಾಗಿ ಓದೋದ್ರಿಂದ ಅವು ಪರ್ಫೆಕ್ಟ್ ಆಗ್ತವೆ. ಪರ್ಫೆಕ್ಟ್ ಆದ ಕ್ವಶನ್ಸ್ ಎಕ್ಸಾಮಲ್ಲಿ ಬಂದರೆ ಈಸಿಯಾಗಿ ಬರೆಯಬಹುದು ಅನ್ನುವಂಥ ದೃಷ್ಟಿಯಿಂದ ಕೆಲವು ಕ್ವಶನ್ಸನ್ನು ಪರ್ಪಸ್ಫುಲಿ ರಿಪೀಟ್ ಮಾಡಲಾಗಿದೆ. ಓಕೆ, ಹೋಪ್ಫುಲಿ ನೀವು ಈ ವಿಡಿಯೋ ಏನಾಗ್ತದೆ ಅಂದರೆ ನಿಮ್ಗೆ ಯೂಸ್ಫುಲ್ ಆಗ್ತದೆ ಎಕ್ಸಾಮ್ ಪಾಯಿಂಟ್ ಆಫ್ ವ್ಯೂ. ಪಾಸಿಂಗ್ ಪ್ಯಾಕೇಜಲ್ಲಿ ನೋಡ್ಕೊಳ್ರಿ ಅಂತ ಹೇಳ್ತಾ ಥ್ಯಾಂಕ್ ಯು ಸೊ ಮಚ್. ಆದಷ್ಟು ನಿಮ್ಮ ಫ್ರೆಂಡ್ಸ್ಗೂ ಶೇರ್ ಮಾಡುವಂಥ ಕೆಲಸ ಮಾಡಿರಿ.